U.S. Senate as National Native Plant Month, which is pretty amazing.
Yay.
I am thrilled to have Dr. Susan Waters with us this evening. Susan Waters is the founder and senior research ecologist at Quamash Eco Research. Her research focuses on plant pollinator community dynamics in Cascadia prairies. Susan's training is in pollination ecology, excuse me, and plant community ecology with an emphasis on species interactions under climate change. He earned her doctorate at the University of Washington, where her research focused on native exotic plant interactions mediated by pollinators and the effects of phenological shifting on those interactions. She currently studies how prairie plant pollinator networks change as sites undergo restoration and as native plant populations rebound. Joining us this evening, we welcome Dr. Susan Waters, who will speak on plants, pollinators, native prairies, and conservation. Thank you, Susan. And everyone, if you would just put your questions in the chat, I will go over them with you all later. We'll ask Susan through me what your questions are.
Great. Thank you so much, Gail. And thank you so much, Denise. I'm thrilled to be speaking to you tonight. I love speaking to the Native Plant Society. It's one of my favorite groups to speak to. So hopefully I can do justice to the great occasion of this month and bring you some excitement about pollinators. As I launch into my talk, I'm going to start by talking about the place where I do my research. I'm an ecologist, and I'm going to use the place to launch into talking about the things that I study, because it's really the place that has rooted me in these questions. And the place is the Puget Trough, Willamette Valley prairies. These are beautiful west side prairies, west of the Cascades. They look like this in this picture, as you can see, in the springtime. And their historical extent stretches from the Willamette Valley up through the Puget Trough region where I live and up to Vancouver Island. They're beautiful and they're charismatic. And they're also very fragmented and have been under considerable threat. These prairies host a whole ream of really unique, beautiful and rare species, including some imperiled species. I've included some portraits of them here. There's the Taylor's checker spot butterfly there. It happens to be laying an egg on the threatened golden paintbrush, 2 listed species for the price of 1. And the streaked horned lark, the Willamette daisy, the Mazama pocket gopher. These are all rare and unique species that inhabit these beautiful prairies. Now, not all prairies by a long shot look like the one I showed you in the first picture. And that's going to be kind of the launch pad for what I'm going to talk to you about tonight. So many of our former prairies, or most of the former prairies in this region, have been degraded or changed from their original extent of native plant diversity via land use changes that have happened largely after the arrival of Europeans in this area. Ranching, fire suppression, invasion of non-native species have all caused tremendous changes in these landscapes. And what you see on the screen here is what I would call a degraded prairie, or a prairie that's been heavily invaded there. That's Hypochorus radicata for you guys who are keeping track at home. It's probably growing in your yard. Luckily, in this region and in this point in time, we have a huge group of very committed people in this area who work incredibly hard to help restore native prairies, to help push these plant communities back towards being dominated by native forb and bunchgrass communities that are more reflective of their historical state. And you can see a suite of pictures here that sort of encompasses many of the actions that are taken in this area for prairie restoration. A lot of this has been carried out by the Center for Natural Lands Management and by the Eco Studies Institute. So that's the context, and we're lucky that all this hard work and ideas and dead ends and retooling and troubleshooting has resulted in prairies that look like a lot of change is happening. Restoration looks like it's working. You can see populations, pretty big populations of really, ecosystems full of native plants with many populations of individual rare plants. You smell the prairie lupin when you walk through, and it looks very beautiful from a human perspective. It looks great from a human perspective. But important questions remain. We're kind of at the cusp of a whole new set of questions in restoration, from my point of view. And that's because for a long time, we've had the underlying idea has been, let's just get these native plants back here. Let's recreate this diversity, and then everything else will fall into place. But the question that really frames a lot of my research and what I'm going to talk to you about tonight is, what about everything else? How does restoration of native plants affect all the other living things that historically occupied this ecosystem? And of course, the other living things that I'm mostly going to talk to you about tonight are pollinators. That's what I study. So before I launch into the pollinators themselves, I'm going to sort of draw you almost literally a diagram, a sort of flow chart that describes how I think about restoration and plants and pollinators. And I'm going to develop this chart, and then I'm going to sort of use it as the framework for the rest of my talk. So hang with me through a little bit of ecologist jargon here as I start. So ecologists in general, or at least I in particular as an ecologist, think of restoration as, we think of it in a different way, perhaps, right? So there's one way to think about it is like the joy and pleasure of having your hands dirty and helping pull Scotch broom, and you're sweaty and you're dirty, but it feels great. There's a sense of accomplishment. You're planting native plants. And I love to do that. I also love to think about it theoretically. And when I think about it theoretically as an ecologist, the way I think about restoration is that it's informed, intentional manipulation of basically three things, at least in my ecosystem. The first thing is disturbance. We have the prairie ecosystem is an ecosystem that evolved in the presence of regular fires. That was an ecological disturbance that helped shape that plant community. That disturbance has been removed since colonization by Europeans. We want to manipulate that disturbance to bring back that plant community. So disturbance is one of the things that we intentionally manipulate during restoration. A second thing that we intentionally manipulate is propagule pressure. And that's ecologist jargon for seeds or bulbs or parts of plants that are going to regenerate. You have a prairie that's been invaded, and maybe you can get rid of all the non-native plants. But even if you get rid of all the non-native plants, it's possible you won't get your native plants back if there aren't enough propagules there, enough seeds in the seed bank, enough bulbs waiting there under the soil. So propagule pressure being the second thing. And the third thing is competition. We manipulate competition by removing invasive plants. So this is how I think about restoration. And when we do these things, we change the disturbance, we change the propagule pressure, we change the competition. We do it on purpose to influence the diversity and abundance of plants, right? That's what we're headed for in restoration. I've made two kind of cartoons of native plant communities. And the first one is here. It says unrestored. And this cartoon shows you a whole bunch of blue circles. Those are meant to represent exotic or non-native plants, and a few green shapes that represent native plants. 3 green squares and a green plus shape. So let's take that for our cartoon unrestored site. There's a lot of exotic plants there and a few natives. We manipulate these things about this ecosystem, and what we hope to achieve is this second cartoon that's a restored site. So in this second cartoon, you can see there are a lot fewer exotic plants. It's fewer blue circles, and we've got more green shapes. So the idea here is that we're going to increase native plant diversity. We're going to increase the abundance of rare species. We're going to decrease the abundance of exotic species. That's what we're hoping to do in restoration. Now here comes the but. But the interesting thing is that we can't do that. All those things that we're changing the disturbance, we're changing the competition. We can't do that and only affect the plant community. We're also going to affect other organisms that live in the system, either directly or indirectly by those actions. And of course, the organisms that I'm most interested in are the organisms shown in this picture here that I couldn't help including. This is a painting by my good friend, Mary Lee, of a bee. So, excuse me, the other organisms' diversity and abundance that we're going to affect among those organisms are pollinators. So let's talk about pollinators for a second. Pollination is a mutualism. It's a symbiotic relationship. So you have a flower, you have a bee or another pollinator. The one visits the other. And in theory, you get a reciprocal benefit. They both get something out of the deal, right? The pollinator gets nutrition. The plant gets help reproducing. The pollinator is carrying around its pollen, right? So they both benefit. The fact that they both benefit means that pollination is really important as a process that's both going to be influenced by restoration and in turn influence restoration. Pollination is really important. The best estimate that we have, which comes from a researcher, an estimate made by a group led by a researcher named Jeff Ollerton in 2011. Our best estimate says that roughly 85% of land plants depend on pollinators in order to reproduce. That's a really, really, really high proportion. And that includes many of our native Pacific Northwest prairie forbs, including every single one I've shown you in this picture. And just because you guys are you guys, I'm going to tell you what these plants are. In the lower left-hand corner is Cascadia hispida, and that's being visited by a beautiful, big, gorgeous Bombus californicus queen. It's California bumblebee. And then up in the left-hand corner is some dogbane being visited, I think, by an andrenid bee. In the middle is Armeria marittima or sea thrift. There's a flower beetle on there. And then we've got Oregon sunshine being visited by a sweat bee, and down below is camas being visited by a fly. They're all being visited by somebody. And when we zero in and we look at just our native plants, our special regional, very own local prairie native plants, and we look at whether they are dependent on pollinators, we find approximately the same proportion as that global study. So we've done that experiment where we've excluded pollinators from flowers and allowed pollinators to flowers, and then looked at how much seed each of those flowers produces, if they get to have pollinators or if they don't get to have pollinators. And what we find is, somewhere between 75, 85% of our forbs are pollinator dependent. Can't make seeds without pollinators. So lots of us know that, but we don't often think about what that means in a visual way. And I'd like to try to convey that with this slide. The fact that pollinators are that important means that landscapes like the one I'm showing you in this picture cannot exist without pollination. We don't get to have them. We cannot have this picture, which by the way has death camas and Limatium pugetensis and you can see some camas fruiting We don't get to have this landscape to look at unless the pollinators are there and they're doing something. This is why pollination is so important. So to bring that back to this diagram that I'm developing here, what that means is when we do restoration, we're not just going to affect the plant diversity and abundance. We're not just going to, that in turn is not just going to affect the pollinators, but the pollinators are going to feed back via that red arrow to affect the plants. That means that this pollinator square here is really important to pay attention to what's happening with it. And that's why I'm so interested in it in my research. Furthermore, not just is the plant diversity important, not just is the pollinator diversity important, but the two are going to actually interact and interlink with each other via a community structure. Certain plants are going to interact with certain pollinators are going to interact with certain plants, but not others. That forms what I call a community structure, a set of interactions. And in turn, that community set of interactions is going to inform how resilient this whole ecosystem is to environmental change. And that's really my ultimate fascination and interest and my ultimate research goal. So tonight, I'm going to try to touch on all of these factors in this diagram, and I'll sort of use it as a framework from here on out. I'll start by just giving a little bit of just a little bit of background about how we do this restoration, disturbance, propagule pressure, and competition, just a brief gesture to it. Here's again, that unrestored prairie. This prairie has actually been has experienced a lot of restoration since the time this picture was taken. But at least at the time of this picture, you can see this is a very undiverse landscape, right? It's like hypochorous and non-native grasses. Here are the things that we do here in the South Sound. In the upper left-hand corner, you can see a prescribed fire. So we're restoring that disturbance to the system. Going clockwise, well, sort of clockwise, going Z-wise, the next two pictures are native seed being put into a seeder. And if we skip down to the lower left, those are seeds being planted and then seedlings being planted out. So increasing the native propagule pressure in the prairie. And then the final picture, those are people who have spot sprayers that they can touch to individual tall oatgrass plants. And tall oatgrass is an invasive plant that's strongly outcompetes many of our native plants. So we've got disturbance, we've got propagule pressure, we're removing competition, and we do all this. What is it actually doing? Let's take a look at what it's doing to our plant diversity and abundance. Well, first of all, we get much increased native plant diversity. This photograph came from a highly restored site. Just even if you don't know the particular plants in the picture, you can see at a glance that there are lots of leaf shapes. There are lots of flower colors in this picture. There are different leaf sizes. If you'd like to know what some of the plants are, there's some balsam root, deltoid balsam root, I guess would be the common name. Leaf there, there's some blue-eyed Mary, there's some western buttercup, there's some sickle-keeled lupin, some native chickweed, some, let's see, what would graceful cinquefoil be the common name of that. So many native plants. In fact, I'm not sure I can even find a non-native plant in this picture. Anyway, so it looks good. We do these actions and we really do get much increased native plant diversity, and it's beautiful. You should definitely come visit. Early May is the time. And if we want to look at actual data, we can certainly provide that too. So here's a little bit of data to start us off. This comes from 3 prairie sites that are being restored. Bald Hill is in Corvallis, Oregon. Smith Prairie is on Whidbey Island. St. John's Prairie is in Portland. The y-axis of these graphs shows floral richness. And if you're not familiar with that term, richness is another form of ecologist jargon that just means number of different kinds of flowers. So number of different species of flowers. And red here is in restored units. and blue is in adjacent unrestored units. And you can see across the board, doesn't matter which year, this is 2 different years, there's always way more floral richness, way more kinds of way more species flowering in restored sites. So absolutely, we get higher floral richness. We also get higher abundance of rare plants like this beautiful golden paintbrush. So for sure, we take those restoration actions, we definitely get changes in plant diversity and abundance, and we get desirable changes in plant diversity and abundance. So now let's start to move over to the pollinator side of the story, which makes up most of this talk. Let's look at the pollinator diversity and abundance. Okay, there's a fair amount going on this slide. I'm going to start with the graph. This graph shows you floral richness on the x-axis. So that's the number of different kinds of flowers. Shows you pollinator richness on the y-axis, the number of different kinds of pollinators. You can ignore the shapes, they don't really matter, but the colors will tell you that some of the sites were restored and some are unrestored. And the line with its confidence intervals shows you very clearly that as floral richness increases, pollinator richness increases. Now, that may seem like a self-evident relationship there, but I got very excited about this graph. And part of the reason is that this underlying assumption, if you build it, they will come, has really been around for a long time. And we don't know that for sure. We don't know where all of the pollinators that used to inhabit these prairies are. Where are they? Are they in refugia? Where did they spring from? We'll never know whether we get back the original pollinator diversity, but we're definitely getting back significantly more pollinator diversity as we restore. And that's super exciting. So positive message number 1 for tonight. Pollinator diversity is increasing with restoration. So that's great. Over to the left of that figure, you can see a list. Most of the things on those lists are, each of those names, for the most part, is a genus. So not species, it's just a list. There's a few that are not genera, but most of them are genera of flies and bees. So it's a list of genera showing you there are a lot of genera of flies and bees showing up at our flowers on the prairie. If I were to list species, it would be hard to fit them on this slide. There's a lot of diversity. Moving down to the picture of the bee faces, I hope you guys all realize that we have shiny blue bees in our native Northwest habitats. And if you didn't, now you know, and you can watch for them. These all belong to a genus called Osmia. You may know Osmia in the form of the blue mason bee, which is just one kind of Osmia. The blue mason bee is the one in the uppermost left-hand corner there. And then we've got a series of mug shots of a whole bunch of other Osmias, to the point that actually we can't identify most of these to species, because guess what? There's so much Osmia diversity that the key that exists for Osmia is totally useless. So we have to identify these to morphospecies. In other words, group them by the characters that we observe and keep records of them, as is shown to the right there. So we have photographs and descriptions that help us group them into these morphospecies, and eventually we'll be able to have taxonomists identify them to species when those keys are revised. This is the place for me to pause and say the person who took all these close-up pictures of these bee faces and bodies is the fabulous David Kappert. You'll see his name throughout my talk. He is a collaborator of mine. We work together mostly in the Oregon prairies. And I'm deeply, profoundly indebted to him for the majority of the good photos in this talk. And I'll return to that name again in a little while and show you a way to look at some more of his photos. So many thanks to David Kappert. So lots of pollinator diversity. Who are the pollinators doing this? Well, it turns out that bees and flies are dominating flower visitation in our Cascadia prairies. I'm showing you the same 3 sites again. The red is bees, the green is flies, the other colors are other groups, Lepidopteras, butterflies and moths, wasps, beetles. You can see though at a glance, most of the visits that we have observed, which is what's on the y-axis, the proportion of our observations, most of what we observe is bee visits followed by flies. So that's really cool. And it's something that we didn't know before, how important the flies were, and how relatively little we're seeing Lepidoptera is a little bit of a concern. But that's what we see out there. If we zero in on the bees, we can identify the bees into three rough groups: bumblebees, which are native social bees; honeybees, which are non-native social bees; and solitary bees, which are a mixture but mostly native, and they're solitary. If we take these three rough classes of bees and we look in the restored and unrestored sites, again, red is restored, blue is unrestored. You can see in every case, at every site, more visits to flowers at the restored sites. So I would say that we have pretty clear evidence that restoration is in fact benefiting pollinators. So that is a really great first step. Not only that, but I just want to pause and return to the plant perspective for a minute, just to look at our rare plants and note that of the rare plants in these sites that we care about a lot, I'm showing two of them here. On the left, golden paintbrush, or Castalia levisecta. On the right, Erigeron decumbens, which is the Willamette daisy. I'll start with golden paintbrush. Golden paintbrush is extremely pollinator dependent. Plants can vary in how pollinator dependent they are. Many plants will make more seed if they get more pollinator visits, but some plants will make no seed without a pollinator visit. Golden paintbrush is one such plant. If you look at the graph down below the golden paintbrush photograph, that shows you the results of an experiment where we allowed bees access to the flower that's open, or excluded bees from accessing the flower that's self-pollination only, which is on the right-hand side on that x-axis. The y-axis shows you how much seed was produced, and that line in the middle of the bar is the average. So you can see that the one that was open to bees made about, on average, produced about 60 seeds. The one that self-produced 0. And this is an average across multiple treatments. So totally pollinator dependent. Yet another reason why we have to think about pollinators in our restoration, because we need to make sure that we're bringing back pollinators that are going to be servicing these rare native plants. Turning to Willamette daisy is not nearly as pollinator dependent as golden paintbrush is, but what we definitely find with Willamette daisy is that in some sites it's pollen limited, meaning that if you give it extra pollen, it will make more seeds, meaning it wasn't maxed out by the pollination it was getting. Okay. So that's an interesting thing for us to know too. And sometimes we might want to know, well, which sites is it most pollen limited at? Are we concerned about reproduction at that site? As a total side note, if you look down at the scanning electron micrograph on the bottom, this is actually not related to pollen limitation, but I couldn't resist including it because it's so cool. This is a scanning electron micrograph of the stigma, or female receptive part of the flower, made by Savannah Richard. And I'm going to see, I think you can see my cursor. So this bumpy thing is the surface of the stigma. This beautiful structure right here, that is Willamette daisy pollen. It can grow a pollen tube and fertilize, allow this plant to reproduce. These two characters over here, those are imposters. Those are other plants' pollen, other Asteraceae pollen that have also been transferred to this stigma by a pollinator. So pollinators are not always perfect for plants. Okay, but take-home message is that many of our rare plants are really benefiting from these pollinator services. Talking about Willamette daisy is an opportunity for me to take a little detour purely to show you David Cappert's marvelous photos and to illustrate the incredible centrality, or if not centrality, at least incredible importance of one endangered plant in terms of provisioning an incredible diversity of pollinators. I'm going to show you 3 or 4 slides. Every picture on these 3 or 4 slides is Willamette daisy. In every picture, there's a different pollinator visiting Willamette daisy. On this page, we have all different sweat bees, halictid bees. They all belong to the family Halictidae. You can see they're covered with pollen, sticking their little faces into these florets that stick up from the flower. Here's some more. These are all cuckoo bees. The top 2 are Coleoxys. The bottom left, I think is Sphacodes. I can't actually remember. The one on the right is a nomada. These are all bees that parasitize other bees. They lay their eggs in other bees' nests, just like a cuckoo bird. Here's a whole other set of bees. The top left is a longhorn going clockwise, a Melisodes. The one in the lower right-hand corner is a leafcutter bee. The one in the lower left is a carpenter bee. This is a lot of diversity for one plant to be supporting. And that's before we get to the flies. Here are the flies. That first one, I'm not sure if that's a sarcophagid or a muscid, but the other 3 are flower flies. They belong to the family Syrphidae. You may or may not have realized that many of our flies have bee-like coloration that helps them, helps potential predators think at least twice before preying on them. And we have an incredible diversity of flower flies in our prairies. The first year we did this, we saw 19 genera of flower flies. 19, that's 19 genuses, not, and multiply that by species within each genus. In case, just as a total aside, if you're watching an insect on a flower and you'd like to know whether it's a fly or a bee, you can look for 2 clues. One is, see those round, funny antennae with the little hairs coming off them? Those are fly antennae. Bee antennae look nothing like that. And also, the fly eyes touch, the bee eyes don't. So anyway, all of these insects are being supported by an endangered plant. Last page, this is Lepidoptera. This is a moth that is mimicking a bee and a couple of butterflies. So these are all wonderful to look at, and we have many more photos at my website, www.quamisheco.com, on a page called Pollinator Gallery. And this is just a little plug for David. If you go to the website and follow that up, you can go to additional sites that David himself hosts with his incredible insect photography. He's a really amazing photographer and is deeply involved in iNaturalist too. So plug for David. Okay, so we have restoration actions. We have impacts on plant diversity and abundance. We have impacts on pollinator diversity and abundance. Now we're going to get to the core of this story and talk about community structure and function, how these plants and how these pollinators interact with each other. I'm going to talk to you using pictures that look like this one on the screen. This is called a plant-pollinator network or a bipartite network. There are different ways to draw it. This is a really common way. And I'm going to explain to you how to look at it since I'm going to show you a lot of them. So first of all, don't try to read the tiny print. You don't need these, and especially don't try to read the tiny print because this is a network that comes from before we actually identified all our insects to species. So you probably don't need to see things like fly 1 and fly 8. But anyway, the way to look at this is these bottom black rectangles here. There are 6 of them, 1, 2, 3, 4, 5, 6. These represent all the plants that were in flower at the time that we did the observations that built this network. So there's little codons that are abbreviations for the plant. Across the top, each of these black rectangles represents a pollinator. If a line connects one of the black rectangles that's an insect with one of the black rectangles that's a plant, that means we saw this insect visiting this plant. The width of the line is proportional to how often we saw that visitation take place. So you can tell a lot by looking at these pictures. For example, in this particular case, you can see there's one plant that's doing a very outsized job in this particular network. Unfortunately, in this case, this happens to be an invasive plant. But we'll come back to the topic of invasive plants and pollinators in a minute. Here's an alternative way to look at plant pollinator networks. And I'm showing you this alternative because I may show you some of these later as well. This is the same kind of idea, but in this grid, the plants are listed on the left side and the insects are listed across the bottom. And you can follow an insect and see, oh, this grid cell is colored. So that means this plant was visited by that insect. So it's just a different way of looking at the same thing. I love to use networks to study plant pollinator interactions for several reasons. First of all, it's looking at a whole community. It's not just looking at one species or a pair of species. It's looking at a whole community. It allows you to visualize patterns of interaction that can tell you a lot that might become relevant to conservation actions or management actions. You can also analyze metrics of the network itself that tell you things about potential stability and resilience of the system. And you can do practical things like view pollinators, the pollinator of a rare plant, what other plants does it visit, that kind of thing. So it's a really powerful tool. And in order to show you some data relevant to this tool, I'll tell you about how we collected the information that we need to build those networks. So I'm going to show you data that come from 6 sites. The sites span 0 to about 12 years of restoration, including a reference site. That's a site that was never grazed and didn't lose fire to the same degree as the other sites. So it's higher quality prairie. We visit each site every couple of weeks. We count flowers with quadrats, as I show across the bottom. So we get the abundance of all the native things that are in bloom and exotic things that are in bloom. And then we do pollinator observations at every flowering species. We randomly choose 2 patches. And we collect or identify every single insect that comes to visit a flower in that patch. Now we have done this for 4 years. And here is a sample result. So this is a prairie called Glacial Heritage. I'm sure some of you have been here in the South Sound. It's in Little Rock, near Mima Mounds. Again, plants across the bottom, pollinators across the top. If a line connects the two, then they were interacting. I've color coded this by red for bees, blue for wasps, tan for flies, green for Lepidoptera, and gray for everything else. And you can see a couple things at a glance. You can see, for example, that this is a network that's dominated by bees, very much a bee network, although there's a really important fly showing up in the middle of it as well. So this is a sample result. And one amazing side effect of getting this kind of data is that we now have the largest collection in existence, as far as we know, of pollinators for Westside prairies. And we're working to leverage that collection and do more with it, find out about the traits that those insects carry and how they fit together with plant traits. But back to this story for now. So we have these amazing community structures. So what? I mean, they look really complicated, but who cares? How can what are you going to find out from them? Well, here I'm going to tell you 2 really practical uses for these community structures in restoration and conservation. Here's interesting use number 1. So an interesting use number one for these networks, I'm going to zero in for a second on the federally endangered Taylor's checker spot butterfly. This butterfly, when it lays eggs, its larvae, when they hatch out, they have to feed on very specific plants. They will eat nothing except for plants that contain a certain biochemical constituent that they require. In our area, there are three things that they eat mostly, but one of them is not insect pollinated, so it's not included in this story. The other two are native plants, harsh paintbrush and golden paintbrush. So the butterfly's larvae choose on harsh paintbrush or golden paintbrush in order to develop. And as a result of needing those host plants, lots of time and money and energy is spent to establish these host plants because we're rebuilding habitat for the butterfly. Okay, so in order to have the butterfly, we have to have the host plants. However, guess what? Both of these host plants are 100% pollinator dependent. They have to have pollinators in order to set seed. And beyond that, not only do they need pollinators, when we go and watch them, pollinator visitation, especially to the harsh paintbrush, is very low. It's hard to even to capture any insect visit to that plant, which is fascinating. So we need these plants, we need them to reproduce, we need them to be there for the butterfly, but they need the pollinator, and the pollinators are not showing up super much. So we pose the question, okay, butterfly needs the golden paintbrush needs the bee needs probably not just golden paintbrush, because bees live for a lot longer than any one given bloom usually. So we pose the question, what other plants support the pollinators that support the butterfly host plants? Well, we can answer this question with our networks. Here's the network, again, plants across the bottom, insects across the top. I've put golden paintbrush over its little black rectangle there. And if you look closely, I don't know if you can see, but there are only 2 lines coming out of that little black rectangle that is for golden paintbrush. And that means that in the whole season of observing there, we only ever saw 2 insects visiting, 2 kinds of insects visiting golden paintbrush. So let's track one of them. Here is a fairly common pollinator of golden paintbrush, Bombus californicus. But at this site, you just have to trust me that I know you can't probably see these individual lines, but just trust me that California bumblebee itself, in turn, only was detected visiting 2 plants at this site. One was this plant that we care a lot about, golden paintbrush. What was the other one? Let's find out. Well, the other one was a plant that doesn't even bloom at the same time at all. It doesn't even overlap in flowering time. That's Prunella vulgaris. So it's the native one. So we have 2 plants that don't bloom at all at the same time, linked by a bumblebee that visits both. And that suggests something important for conservation. It suggests that plant species that flower at other times, like this prunella, may be important to supporting the population of the pollinators that in turn we want to have show up next year to pollinate the plant that we care about, the golden paintbrush. So this is one practical use of these networks. This is not a result really, it's a hypothesis, but it's a practical stepping stone for getting to something useful in conservation. Here's another example. Here's golden paintbrush at another site. At this site, we had 3 different kinds of visitors. The tan one is a fly that probably does a terrible job of transferring pollen. You just have to go with me on that based on this particular fly's life history. It's not likely to be a great pollinator. Let's follow one that is likely to be a good pollinator. This is the yellow-faced bumblebee. Super common, super common, super common. It's probably the most abundant bumblebee in Seattle, but at this site, We only ever saw it visiting 2 species, which was very surprising. One was golden paintbrush. What was the other one? The other one was a ubiquitous weed. Okay, this is a really interesting result. This is a really interesting implication that we have to be careful about. So the implication here is, gosh, maybe we shouldn't entirely eliminate this weed until we have other floral resources in place for this bumblebee, because we need this bumblebee to survive from year to year in order to visit the plant that we're trying to conserve. So this is an interesting and perhaps slightly controversial for some people idea. Now, I would hasten to say that with this particular plant, Hypochorus radicata, we're not in any risk. There's no risk that we're ever going to be eliminating this plant. It's not a worry. But there are many plants out there that people are, perhaps spraying to remove or getting rid of them en masse. We have to remember that those are resources that our native animals have learned to use, and that we have to be intelligent about the way that we get rid of them gradually rather than all at once. So that's one way that we can use these networks. Here's a second way. We can ask, well, gosh, are we restoring plants that support diverse pollinators? So restoration takes years upon years in these sites. 15 years into it, maybe we feel like we're getting there, right? That's a lot of years. There's probably plants, different native plants being seeded in or plugged in year after year. They don't all happen at once in a single year. If we knew that we were choosing early on in that 15, 20-year process, choosing plants that support a wide variety of pollinators, then we might be helping support a diversity of pollinators so that those diverse pollinators are in place by the time we later plant more specialized plants that require a particular pollinator, right? So we could potentially help us support pollinator populations to support more specialized plants. And we can do that really easily. And all I've done here is to put circles around plants at these 3 networks. These represent 3 different pretty highly restored sites. I've put circles around plants that have a lot of lines coming out of them. Like a lot of different insects are visiting them. When we do that, we come up with some of the usual suspects, chemists, Oregon sunshine, and what is the common name? Short spur rosy plectritis. I can't remember the common name. Rosy plectritis down there. These are all plants that they're already everywhere or we already restore them. So no big surprises there. But then we come to a couple of other ones. Potentilla gracilis turns out to be an incredible pollinator plant. Micranthes integrifolia on the bottom there turns out to support a whole host of wasps and beetles that nothing else is supporting. So we've suggested to some of the managers, what about introducing these plants early in the restoration process? Maybe that will help support pollinators that will in turn support other plants. And several of the managers in the South Sound have acted on this and are now including these plants regularly in their early cycle. So there's another practical use of networks. Now, what about invasive plants? Like lepidium, that's field pepperweed, oxeye daisy, and crepus in this picture. So some of these guys are also visited by diverse insects. So how important are these plants to native pollinators? Well, we can use our networks to get a glimpse of that and to develop a potential hypothesis about that. And one way we can do that is to do simulations using the networks. We can simulate what would happen if we really were able to get rid of these introduced species that we're trying to get rid of. I'm going to show you the results of 2 simulations. What these simulations do is they use the plant pollinator networks and they remove each forb one at a time. They remove each plant and then they predict how many pollinator species are left, basically. And you can arrange it so that they will, the way the simulation works is it will one by one remove every plant at the site. And the way I've arranged it here is to just make sure that the first ones that it gets rid of are the non-native ones. Okay, so I've chosen to do this at a really diverse site called Johnson Prairie. It has 7 dominant non-native plants listed there. And the figure here on the x-axis shows you the proportion of plant species that have been extirpated in the simulation. And then on the y-axis, the proportion of pollinator species left. Now, of course, if you let it go all the way to the end, it goes to 0. But what we really care about is the first 7 plants. And if we look at those first 7 dots, what we find is that we lose about 10% of our pollinators if we get rid of every single one of those non-native / invasive plant species. So this is a pretty interesting result. I mean, 10% is, that's a pretty big proportion. But guess what? If we do it at a less restored site, like Cavness Ranch, well, at Cavness Ranch, we only have to extirpate 4 non-native species. But since there's so few species at Cavness Ranch, when we get rid of those 4, we lose about 18% of our pollinators. This is predicted. This is not reality. This is predicted. It's a simulation, but it's a very interesting result. And the conservation implication here is it's really important to provide alternative native flowers when you're fighting weeds. It's the same implication as in the previous study. Now, this doesn't mean we don't want to get rid of these invasive plants, because we do, right? But we need to pay attention to how we do it and the impact on pollinators, and think about scale. You know, getting rid of all the blackberries that are over here in the corner of the field is probably trivial if you have 5 other fields of blackberries. On the other hand, if you're getting rid of the blackberries and you're in the middle of Seattle and the blackberries are the only floral resource for a mile around, that's a different thing. Okay, now, underlying all of this though, there's a really big assumption, and anybody who knows pollinators is probably already saying, hey, but wait a minute. The really big assumption here is that the pollinators aren't changing their behavior, right? That if you remove some plant, they don't just switch and use a different plant. Some pollinators can, some pollinators can't. Lots of them can though. So this is actually an unanswered question that I'm very interested in answering. And a student who worked with me, Amber Haley Kinnaird, did a little pilot study on this. Her question was, well, how does pollinator behavior rewire networks when you take away an invasive plant? And what she did was before and after networks. So again, plants across the bottom, pollinators across the top. Here's the network, and it had this invasive plant, Hypochirus radicata, in it. And here's that plant supporting a whole bunch of pollinators. And what we did was we got this network data, and then we clipped off every single Hypochirus flower head, every capitulum in those plots. It was a lot of work. And then we surveyed the sites again. And upon removing Hypochirus, you can see it's completely reorganized, right? You can see the red bee, for example, is visiting things in a totally different pattern than it was before. We saw connectance of the network increased. We saw diversity of pollinators increase for 2 native plant species. So this is a really exciting little tidbit that we're hoping to build on to get a sense of how restoration can influence communities. Okay, I'm running a little bit late here, so I'll try to finish up quickly, but I want to get to the good part at the end here, which is the little snippet, the little whiff of how maybe we can relate all of this to resilience, to environmental change. For a moment, I'm going to run through the 6 sites that I told you lie on a restoration gradient. Here's the site that has been restored for 0 years. This is the site that had been restored for 2 years. says these are different sites, right? So a series of sites. This site had been restored for 5 years. This site had been restored for 8 years, 11 years. This was our reference site. Okay. You don't have to know anything about fancy algorithms or interpreting these networks to look at that and say, wow, things are getting more complex. Pollinators are returning. You can see there are more pollinators across the top. You can also see there are more plants across the bottom, but it's not just that there are more of each, it's that the interconnections and the way that they're interconnected is changing. So this is really exciting. This was sort of the first year's result was, okay, if we look across these sites, we can see that there's differences. But now we are at year 4, and finally, for the very first time, we're able to actually trace sites across 4 years and see how they're changing through real time. Okay, and this is hot off the press. Nobody's ever seen this before. You're the first. I'm just going to show you 4 networks at one site. This is a very degraded site. This is in year 1. You can see almost every pollinator was being supported by this one plant, which is a native plant. Look at what happened in year 2. Wow. So this is the beginning of restoration. So they came in, and the process is a little bit, that this process of clearing everything away and starting again with a native plant community is disruptive intrinsically. So in year 2, boy, that network went to next to nothing. Year 3, wow, okay, this is after they had burned and seeded. Big rebound. Year 4 looks a little bit, perhaps a little bit less complicated, but that's possibly because we had one of the flowers that didn't flower this year. Suffice it to say, we're seeing some real rearrangement through time. We're seeing different players, different plant players prove to be key. And we're actually seeing some plant players change their roles as restoration goes on. So stay tuned as we dig into the details of what's happening at each of these 6 sites over time. I'm very excited to get to that. And the very last thing I'm going to give you as a takeaway before I draw to a close is that we're also working on being able to talk about how good these networks are. And by good, I mean, how close are these networks coming to approximating the ecological processes that we think of as being important, like specialization, diversity, et cetera. I guess those are qualities, not processes. This will be a little hand wavy. If anybody wants more details, I can explain it more afterwards. But rather than get too far into the nitty gritty, I'm going to just tell you that there are metrics of these networks that can be measured, that measure the ways that things are connected. For example, you can have lots of little subgroups that are loosely connected to each other, or you can have everything that's kind of connected to everything else, right? And a lot of different in-between kinds of characteristics. 2 of the main metrics that I'm interested in are modularity. That's how many little subgroups you have that are interconnected. And what we expect is what's shown on this screen. The x-axis shows from unrestored to late stage restoration. What I expect to happen is that as restoration goes on, modularity, or organization into lots of little subgroups, is going to increase. And that's because I'm expecting plants and insects to behave in a more specialized way as we add diversity. By the same token, I'm expecting a different metric called nestedness to decrease as restoration goes on. And that's because nestedness often reflects what happens in a network when you have a super generalist that visits everybody or is visited by everybody. And as we get rid of our invasive plants, I predict that we're going to see less and less nested networks. These characteristics, modularity and nestedness, relate to the concept of resilience because they influence how a network will respond when a species is lost, right? You could have a network that when a species is lost would just collapse. And you could have another network that when a species is lost is able to just keep on functioning because of the way it's organized. So to date, we have only 2 years of results as far as these characteristics of networks. Those are shown in these pictures. The blue is, let's see, red is 1 year and blue is the other year. I can't remember which is which. But you can see the purple shows you the hypothetical relationship and the blue and red show you the actual relationship that we've detected in the 2 different years. So stay tuned for more news on whether restoration is actually promoting resilient, friendly structures in these community networks. We hope to be able to tell you more. So, and with that, I will just briefly talk about future directions. We're hoping to look at plant pollinator networks across the range of the prairies, from all the way down south up to up north, looking at floral and insect traits in networks that shape these interrelationships. We're also hoping to track the effects of invasion by an introduced bumblebee, which will be a first for our region. And of course, we're going to continue to use networks in conservation of endangered plants and insects. So with that, I would like to thank too many people to possibly do justice to. First, great thanks to our funders. Jim and Beerta Falconer have funded the past 2 years of this project, last year and this year, and they have made it possible to continue this project into a multi-year state that really is going to give us some ability to say something about these prairies, and I'm super grateful for that. The initial funding for the project that really launched it came from the U.S. Fish and Wildlife Service. I'm really, really grateful for that. Army Corps of Engineers has supported the project at Oregon sites, and the DoD has supported our ability to work on JBLM prairies. The pictures show reams of incredible people who have contributed to the project. They're listed over on the left, and of course on the bottom there, those are our other collaborators and folks who have helped with taxonomy. And with that, I'm ready to take your questions. Thank you for listening.
Susan, thank you. That was just wonderful. I've heard you give various talks on the prairies, but every time I get more and more intrigued. So, oh my goodness, thank you. So I'm now going to present some questions. Nathan, who was working on his own on a prairie project, asked, how was the plant pollinator interaction network constructed? Is there software available or some other method? And then he clarifies his question. It is regarding non-grid type of network.
Yeah, so you can build these networks using R. You code them in R using a package called bipartite. So R is a freely available software that's used by all kinds of people to do all kinds of analyses.
Okay, thank you. Anonymous attendee asks, pollinator inhibitors such as pesticide, herbicide, urban and monoculture have been shown to reduce pollinator abundance. Does floral diversity assist in counteracting these pressures?
Oh, that's a great question. Wow, that's a great question. So no, not necessarily. Let me rephrase the question and make sure I'm understanding it. I think I got most of it, but I think what you're asking is, can you counteract the effect of pollinator declines that may be influenced by the use of pesticides by providing more food to the pollinators, basically, by providing more habitat? So I guess the answer to that question is really, it's a little bit of a mixed answer. So on the one hand, if you have pesticides, heavy use of pesticides and you have suppression of insect immune systems, you have real internal health effects. You're not going to solve that fully by providing more habitat. On the other hand, pollinators that have a better nutritional status because they were able to forage on a wider variety of flowers, some of which provide not just nutrition, but also, you might say, probiotic benefits. Not even kidding.
Yes, I'm sure.
Those pollinators that are able to have a better diet are also better able to deal with the stressors in general in their lives, up to and including pesticides. But of course, you can, once the pesticide impact sort of crosses a threshold, then you can't rescue things by providing habitat. I hope that made sense.
And some of this may be answered. Doug Talmany is giving a talk at 10 o'clock this coming Saturday. If you want to register, he's going to give a talk on the little things around the world. And he's going to address monoculture and other items that you ask about. And
yeah, certainly monocultures are that is something that we can solve by planting gardens and providing more habitat for sure.
Yes. Another question is there a place to buy seeds to be able to plant these plants in our own communities, thinking to help eliminate the island effect on extinction?
Say the second part again about the island effect.
Thinking to help eliminate the
Oh,
island effect on extinction by buying more.
right. Oh, great thinking. So in terms of buying prairie plants in particular, there are a couple of different suppliers of seed. I usually interact with those who are supplying seed on a large scale for restoration rather than on a small scale for individuals. So my knowledge is not super up to date on that. I know that, there's like Fourth Corner, there's what are they called in Oregon? Is it Heritage? No, that's somebody else. I'm sorry, I would probably have to look up the specifics and get back to you. But one thing I will say is that I have, I spend my summers walking through these prairies all day long, every day. And when I walk through giant patches of things in seed season, sometimes I have no qualms about taking a little handful of seeds and putting them in my pocket because there's countless seeds going onto the landscape. And I throw them in my dooryard in the hopes that they will germinate in my front flower garden. And many of them do, but the best ones, the easiest ones, I'm telling you, are the annuals. And that plectritis, the rosy plectritis that I showed you earlier, that is a fabulous pollinator plant. It's really easy to grow. So I highly recommend plectritis.
Becky wrote in quotes, in capital letters, excuse me, that was really cool.
Great.
Larissa from Victoria, British Columbia asked, do you have any advice for starting pollinator monitoring with a volunteer group?
Oh, wow.
Victoria, BC. We would love to include this type of monitoring in our campus. Gary Oak Ecosystem Restoration Project.
Mm-hmm. Yeah. Well, you guys have great experts up there. I know Dr. Sandra Gillespie is speaking in this very series later on, and she's from up there and has done a lot of work in those very prairies. So she would be a great resource. But also I'd be happy to talk to you about it. You can email me at the email that's on the screen there, and I can respond with more.
Okay, space. Becky Stinson suggests for seed inside Port Townsend.
Oh yes, yeah.
And I'm going to go to some of the chats now. Deb Naslin, who is the president of South Sound Chapter, says, thank you, Susan. Fabulous and fascinating presentation.
Thank you.
Lisa questions. You mentioned that you're seeing few Lepidoptera pollinating in some of these sites, and that's a bit concerning, but it also seems like there were very few wasps in the diagrams. Is that just because wasps are always the lowest number of pollinators? How do you boost Lepidoptera populations in these prairie projects?
Yeah, great question and great
Yeah.
observation. So yes, so Lepidoptera are butterflies and moths, and we see them at way lower rates than I would have expected. Keep it in mind that while we're observing, you know, we're standing in front of a patch and we're focusing all our attention on like this patch of camas for 30 minutes, you know. So if something flies by in that patch of camas over there, we don't necessarily see it, right? So, you know, you have to take it with somewhat of a grain of salt. Still, we've been out there for 4 years. We observe we observe every single thing that comes into bloom. You would think that we would see more than we do. I can't explain why we don't. I mean, I just think it concerns me. I think that the Lepidoptera numbers are low. And that's maybe a topic for the Washington Butterfly Association to talk more about, and others who know more about Lepidoptera than I do. In terms of the wasps, there's a good reason why wasp visitation to flowers should be lower than the other insects that we see, and that's because wasps are predators. Most of their diet comes from capturing other insects and eating them. They supplement their diet by drinking nectar from some flowers at some times of the year. And so it's not surprising that we don't see them very much. It's actually interesting to me that we see them as much as we do, because they probably play a little bit more of a role than we would have thought.
Okay, Susan says this is an amazing amount of work. Fantastic presentation, and I'm so glad I got to see your work. Thank you. Let me try to scroll down here a little bit. Lionel says, great presentation of complicated but interesting and important subject. Jennifer Lynn, thank you so much. Susan asked another question. She had made a comment before. Are all the people who work on this study students, volunteers, or paid researchers?
Oh, great question. The people who work on this study have ranged from, so I am the primary investigator, and then I hire techs, but then I also collaborate with other folks. So there's a project in Oregon headed up by Tom Kay of the Institute of Applied Ecology and his team. And then the photographer I mentioned, photographer and entomologist, I mentioned David Kappert, who is sort of part of that team. So there's, the project has drawn in a lot of people from a lot of different arenas. The majority of people who are out doing the field work are paid techs, so field techs. Many of them are usually they're college students who had just graduated within the last year or two, typically.
Okay. Rob asked, do you have a recommendation on how to collect Plectritis sea blush? It seems to ripen and then drop. And he says he's also adds, I'm working on 2-acre prairie restoration creation in an urban area that was historically exotic grasses. Do you have a recommendation on high impact species that should be included, or should we aim for a wide diversity of flowers over the season?
Great questions. Let's see. In terms of wide diversity, in terms of the restoration, I always recommend for people to try to go for diversity and a range of phenologies. In other words, a range of flowering times, because we have, we have things that come into flower and go out of flower and come into flower and go out of flower. And through all of that, we have pollinators whose lifetimes, or whose activity times last more than the bloom of any one flower, right? So we want to cover the season. And then the Xerces Society is a wonderful resource for looking up, you know, native plants to use for pollinators. I would definitely recommend them. Xerces, Xerces.
Okay. Emily asks, can you recommend any resources to learn to identify different species of local pollinators?
Oh, yes, I can. As a matter of fact, there's a fabulous brand new resource. And you know what? I'm gonna just, I think I can get it up here and then just share my screen with you. The resource comes from the Washington Native Bee Association. And it's called WashingtonBumbleBeeID.org, WashingtonBumblebees.org. I'm going to share my screen for a second here, if I can figure out how to do it. Share screen. This is the site. It was recently put up by David Jennings and colleagues, and it shows you how to identify bumblebees by color. It also has a dichotomous key. It's got a lot of great information, and they're very friendly people who answer questions. And while I'm showing you online resources, I'm also going to just jump over here. This is the pollinator gallery at my own website. These are all David Kappert's photographs, and you can see that it goes well beyond you know, the tiny tidbits that I showed you. You know, we got flower spiders capturing bees and all kinds of stuff. So great resources to take a look a little further. But WashingtonBumblebees.org it's pretty hard. In addition to that, in terms of pollinator ID, the best thing I could recommend, see if I could lay my hands on this book. This book is not specific to Washington, but it is a really excellent book. I highly recommend it. It's Joseph Wilson and Elizabeth Messenger Carroll, The Bees in Your Backyard, and it's full of natural history. It goes into all kinds of detail on families and identifying characters. So.
Wonderful resources. Thank you. Let me see. There's some more questions here. From SG again, will you be looking at other influential factors of pollinator abundance alongside floral richness? For example, suitable nesting sites for bees?
Oh, yeah. Excellent question. Yeah, everybody always measures floral abundance, and people rarely measure nest site availability because it's really hard to measure. And in fact, no, we are not measuring nest site availability. Partly, however, that's because it's not super likely to be a limiting factor in our natural areas. Like nest site availability, I would think of as being more likely to be a limiting factor in urban sites. But where we are, you know, these are prairies, there's bare ground between the tufted bunch grasses, there's potential cavities for cavity nesters. So I think that my instinct is that nest sites are not a huge limiting factor here. So, but it is a great question, and it certainly is a consideration.
Okay, Denise writes from that you can find the WNPS native plant and seed sorts document online. This document is vetted annually and provides a list of resources available around Washington state for seeds. So if you go to our website, WNPS.org/content, and then go to documents, plants, /, in there. Denise, I don't know if we can post this on a screen and share it so people can see that or not. It's beyond my limited technical ability.
Yeah, I agree. I just want to share, we actually have several resources available about pollinators and of course gardening and many things on our website. The banner on our homepage, which I'll show you shortly, if you click on that, it'll take you to an interactive poster that we've created for Native Plant Appreciation Month that includes our partners. Susan mentioned the Xerces Society. We have a link on their logo. We also have a link on one of our big bombus bees on our poster to the new Washington Bees website, which is pretty exciting. So do take some time to visit that. And I'll just kind of go over, people asked us last year during the NPAM, do we take donations? Of course, WNPS takes donations. We are member and volunteer funded. Thank you, all of you, for being a part of sustaining WNPS over the years. And you can certainly make a donation, or we welcome memberships 365 days a year. So I'll just show you real quick on our website. Let's see how to get to some of
And
those.
there's one more question when you're finished, Denise.
Okay, very good. So some of those gardening resources, if you come to our homepage, this is tomorrow night's program, by the way, how to build a public park. But here, if you come to our plants section and then our gardening with natives section, and over here we have sourcing native plants. Every year we update this document, and it includes several member organizations as well as others that have contacted us to say, we have native plants, we have seeds, please put us on your list. And there's some folks tonight here on this call that I noticed: Robert Pellant from Pacific Rim Institute and Bridget McNasser from Oxbow Native Plant Nursery, both growers and both restoration enthusiasts. Here we have direct seed sales, BFI seed sales, Inside Passage, as previously mentioned, all organizations that have supported our restoration projects. So this is a marvelous resource, even wholesale resources. And we do rebuild this every year to make sure that these organizations are still in business and active. Later on this month, we'll have Mel Asher, who worked for BFI Seeds. She's now taking over Derby Canyon Native Plant Nursery. And Mel will be talking about propagating, particularly on the eastern slopes of propagating for restoration. So remember, our website is a wealth of resource, and I'll just share again, WNPS.org. And I specifically, this link will take you to our beautiful NPM poster, complete with pollinators and a variety of wonderful clickable things, brand new, as well as the full list of Native Plant Appreciation Month activities, of which there are many this year.
It's amazing.
Yes.
It's amazing.
And there is another question, are you, from Janice, and I don't want to miss this one because it's a good question. They're all wonderful, but this one is, are you experimenting with burning timing, excuse me, and its impact on various life stages of pollinators?
That is a great question. That's an excellent question. The short answer is no. But the longer answer is, I guess, what I would say about that is that there are several people around who are doing that. Notably, although not in the prairies so much, notably Lauren Punicio of the University of Oregon, I know, has a project going on with recovery after fire in Oregon and pollinator communities there. Here, the problem, a little bit of the problem with doing that in the prairies is scale. And that's because when a burn happens, it's usually on a scale that a pollinator can just fly over to someplace nearby, right? So it's hard to compare side by side areas, and there aren't that many preserves being burned at the same time. So it's been hard to get replication experimentally, but it is a really great question. My general sense is that because our burned patches are relatively small, that what we get is just regenerated beautiful prairie, and we get pollinators pouring in from the sides to sort of take advantage of the new habitat. But the timing of burning would definitely make a difference in that. And one more thing on that, there is definitely research going on how the timing of burning affects the plant community. So stay tuned.
Wonderful. Just want to add that on Earth Day, April 22nd, we have two wonderful speakers. We have Sarah Hammond's going to be talking on the importance of soils for restoration. And Julie Donald is speaking about native plants and native pollinators for even home gardeners, not specifically prairies. But Susan, you received many accolades of praise for this wonderful talk. And I can't thank you enough from the bottom of my heart for this wonderful talk. It was just wonderful. Thank you so much.
Thank you so much to everybody for spending yet another evening looking at a screen. I know we're all so zoomed out. I'm totally grateful for you all showing up and being interested. And I'm so glad you're out there looking at pollinators and thinking about plants. That's the hope for the world.
Everyone have a wonderful evening. Have a wonderful Native Plant Appreciation Month. Come celebrate with us again. And remember, it's also National Native Plant Month.
Yes.
Our US Senate actually agreed unanimously on something. So that's a first. It's a good beginning. So thank you again. Good night, all.
Many thanks to all of you. Good night.
Good night, all. Thank you. This program has been recorded. Recordings will be posted on our website. You can link from the homepage. Thank you, and we hope to see you back at another Native Plant Appreciation Month program. Good night, everyone. Thank you, Susan. Marvelous.
Thank you, Denise.
Thank you.
Thank you, Susan.
Thank you.